So this is a panel on novel methods and technologies, and it's my great pleasure to have six experts here. Many come from industry. We absolutely want that collaboration between academia and industry, and it's great to see you all here on a Sunday morning. I will share my slides and give brief introductions, and then we have more than 100 slides. So you might have seen my email asking you to stay within 8 minutes, which I think is hard because you have so much cool technology and cool research going on. Dan will support me in having a time already. So there will be some loud noise going off after 8 minutes. And we really want this discussion. And also we can't run over to the next panel because that starts at 11 sharp. So again, thanks for coming in. It's great to see you all here. Sarah, especially, it's wonderful to see you here as a CIFAR co-director. So many things go to CIFAR for financially supporting this event. And we had an amazing setup at 22 hours so far. And we have another 2 hours to go. And then there will be 24 hours total. So I will just share my slides, and you might decide to actually share your own slides. I'm at a lake here, so my internet is not the best. So it's actually better if you want to share your own slides, and that might help you to stay within the 8 minutes. Let me just show you what the program is for this 1 hour. So I'm sharing my screen, so you should see that. We have different companies. So we have Majd from single cell technology. We have Jiang He from Vizgen. I recently had the opportunity to visit, and it was absolutely fascinating to see what's going on there, and also to see how closely these new companies collaborate with academia. We have Colles from Takeda. We have Daniel Moline as a science and technologies advisor. We have Sarah Teichmann. And again, it's a true pleasure to have you here. I know you're very busy at a different event, but you managed to come join us here. And then also we have Tobias Wenzel, and he is one of the young scholars just recently joined CIFAR, bringing all kinds of cool technologies and a lot of expertise to the table. And new fields of science grow by attracting young brains and new technologies. And then with these new technologies, we can see things that nobody could have seen before. So Majid, I know your slides are next. Do you want to run them yourself, or would you like me to run the slides?
I can try to run them, and if I struggle,
please. And this way we also see each other in between, and Dan can start the timer. It's so much nicer.
Let me see. So the first usually 2 minutes are difficult because then you have to figure out how to put the
I see your slides, and we are now
Okay.
on this second one.
What is it? Oh, there we go. So laser pointer. Do you see my laser pointer as well?
Absolutely, yes.
Sounds great. So my name is Majd. I'm a scientist at Cell Signaling Technology, and I'm going to showcase the interesting technology that we released 2 months ago, which enables customers to measure intracellular protein. And because we're famous for signaling pathways, we're showing that how you can measure all of these signaling pathways in your single cell RNA sequencing experiments. Just a quick overview. Single cell RNA sequencing has been in the field for over a decade now. It was used to identify hard to detect cells, very rare cells in a big tissue. And it was used a lot in the field to look at tissue heterogeneity, as well as identify how cells interact with each other and their environments. This has been done extensively in the field. And this is an example to show, this is a UMAP where each dot represents a different cell, a cell, and each color represents a different cell type. And in this case, red represents B cells, blue are T cells, orange are monocytes, and so on. And this is an RNA-based clustering based on the transcript. And what I would like to show is a technology that was developed around 2018, where DNA antibodies were conjugated to DNA sequences, were barcodes, and the cells were stained with those DNA conjugated antibodies. Once the unbound antibodies were washed off, these cells were loaded into a single cell RNA-seq platform, which allows customers to capture the RNA as well as the barcode from each antibody. And this is a CIFAR technology to measure both surface markers at the protein level as well as transcripts in individual cells. Now, you can see that CIFAR has some limitations because, we're missing a lot of the intracellular protein readout in your single cell assay. And this is why IntraSeq was developed. So we worked for a couple of years on this project to make sure that we're fixing the cells, permeabilizing it, and making sure that the RNA does not leak outside the cell and we don't have any RNA degradation, while also enabling an intracellular protein staining using our DNA conjugated antibody. And this is what IntraSeq stands for, intracellular protein and transcriptomic sequencing. And using our CST antibodies, we can measure all of these intracellular events that happens in your single cell assays. So I want to mention that this technology was validated and developed by CST using the 10x Genomics platform in the 3' kits. So what is IntraSeq? It's technically a straightforward protocol. We have an assay kit and also a cocktail of antibodies, which has multiple antibodies in this cocktail. And the protocol is a 4-step straightforward immunostaining protocol where the first step you have to fix your cells, and you can fix and store your cells if you would like to. And then on the next day, add the SC block buffers, which is basically where all the magic happens, where you have to, it's optimized where the RNA is not degraded and is preserved while also obtaining a very good antibody immunostaining readout, because sometimes antibodies have some non-specific staining background, especially when you start fixing and permeabilizing the cells. And because we have a lot of knowledge working with antibodies at CST, we try to optimize the protein signal as well. And afterwards, you add the intraseq conjugates, let it stain overnight to maximize the protein signal. Once you wash, you load it into the 10x Genomics platform. And in total, this is a 1-hour bench work spread out over 2.5 days. So I'm going to show you in the next couple of slides how IntraSeq can help your work and seek what you've been missing, because the transcriptome is just one mode of your single cell. And once you start adding more layers, there's a lot of new information that you get in your single cell RNA sequencing assay. And this is an example where I'm showing TCF1 transcription factor in this UMAP, which, this is a feature plot which shows expression level of TCF1. Blue means low expression, red means high expression. And you can see that at the transcriptome, TCF1 is upregulated in this cluster of cell or the cell type, and it's low in the other cell types. When you start looking at the protein level, you see a much homogeneous expression level of TCF1 in this cell type as opposed to the other one. And it also corroborates what you see at the transcriptomic level. And this is an example here also with NF-AT, which is another intracellular protein. It's upregulated in these 3 regions and it's downregulated in this cell type. And we see the same pattern at the protein level, but you can notice that in these experiments, especially using the IntraSeq technology, that the protein expression is more uniform and it gives you a more accurate representation of your target expression. And that is due to many reasons. And one of them is, transcripts are usually unstable and they're usually prone to degradation. And there's, on average, more protein molecules in a cell compared to RNA molecules. And using IntraSeq, you can get this representation more accurately than the transcripts. And what we found over the last years is that this is something slightly expected, but what we're showing here is a Pearson correlation coefficient, where when it's high and it's 1, it means that the transcripts and the protein highly correlate with each other. But what we're trying to show here is that more often than not, a lot of the intracellular protein, and because we're at CST, we have a lot of antibodies against post-translational modifications. These modifications do not correlate with the transcripts, but it is kind of expected because these modifications happen at the protein level, and so you cannot make a correlation at the transcript. But what we're trying to showcase here is that you can learn a lot of information using our technology because you're looking at these modifications that happen on the protein level, which tell you whether your protein is activated or inactivated. And this is an example here where we took the CD8 positive cells based on a surface staining. And our technology allows a surface staining expression. Once you take these cells and recluster them based on the RNA, we have three different cell populations: naive, memory, and effector cells. And you can see that TCF7 protein is upregulated in the naive state, which corroborates with a lot of previously published studies. TCF is a marker of naive cells. And when you look at TBET or TBX21, it's upregulated and enriched in the effector state. Same with phosphokreb-serine 133. Now, when you start looking at the transcript level, you don't get the same image. And even if you sequence deeper, you will not get the same representation and differential expression that you would see at the protein level. So what we're trying to show is that, with intraseq also, you can characterize a subpopulation of cells and different cell states. I think I have less than a minute left. I just will keep going.
Yeah, 20 seconds.
Okay, I'll try to be quick. And this is an example because we're at CST, we make antibodies against post-translational modification marks where you can measure phosphoproteins, acetylation, methylation. And these marks I like to describe them as light switches on your protein, which tell you whether your protein is active or inactive. So we took Jurkat cells and we inhibited them with drugs. And um, yeah, so these on the right are the drug-inhibited cells with wortmannin, which is a PI3K inhibitor. And if you look at the total AKT protein level, it's the same in both conditions, whereas in phospho-AKT, it's downregulated only in the drug-inhibited cells, which showcases and offers a proof of concept that you can measure PTM, post-translational modifications, on top of your transcript in your single-cell RNA sequencing assay. And these marks tell you whether your protein is active or inactive in your single-cell state. In your single cell protein assay. Because you're measuring transcripts, what I would like to say that, you can correlate these phosphomarks with different transcripts and see what's upregulated and downregulated. And in this case, because phosphostat-3 acts as a transcription factor, if you know which ones are the direct targets of phosphostat-3, you can actually get the whole mechanism of the in your experiments, whether you have 2 or 3 different conditions, and you can get the direct mechanism in different cell types in different conditions.
Majd,
So,
I'm going to cut you off here because
and
this
yeah,
is just in the interest of being fair to everybody.
that's
So
fair.
thank you so
Thank you
much.
for your
We
time.
will share those slides so everybody will have access to the full story. And of course, you could talk much longer.
Yeah.
The next expert on the panel is Jiang He from Vizgen. And again, great to see you here. Great to see your environment. And please try to stay within 8 minutes. There's a little timer in the top right, thanks to Dan here on the call.
Yeah. Thank you so much, and really pleasure to be here and sharing some of the work that Vizgen is doing. As a brief introduction, my name is Jiang. I'm a co-founder of Vizgen and VP of R&D and reagents. So today what I'm going to share is what Vizgen is doing and some of the new product that actually Vizgen is launching. So I guess this slide really doesn't need a further introduction since we are talking a lot about cell atlas and with a biological system made of so many different cell types intricately organized in 3D and interacting with each other. Spatial transcriptomics is really points to sort of profile the molecular signatures and features of individual cells. And with that information, we're now able to very easily map out all the cell types, catalog them, and understand how they interact with each other. And this is essentially the spatial genomics field is all about. And as a company, Vizgen is one of the first to bring a commercial spatial transcriptomics platform at single cell resolution to the market. And Merscope builds based on a Murphy's technology, which is a RNA fish imaging technology. It allows us to image hundreds to thousands of RNAs in a single run with extremely high sensitivity and accuracy. And we can capture up to, now actually this is a little bit inaccurate now with the new product, up to 3 square centimeter of tissue size and 100 nanometer resolution. And there's no need for sequencing here. And as mentioned, Merscope was built based on the foundational Murphy technology, which was developed by Xiaowei Zhuang at Harvard University. What this technology really is about is that it's sort of a combination of single molecule fish and preparatory barcoding system where it really makes it possible for us to do direct in situ RNA imaging at a much higher plex level. And this is a platform solution so that it's a system in one, it's a easy to use solution, fully automated system in one go. And to do MERS-fish or MERS-scope measurement, it begins by designing a gene panel, and Vizgen allows full custom gene panel design. You can choose whichever gene you want, but at the same time, we also offer some predefined gene panels. And with the gene panels and other associated reagents, then you can start to stain the samples, and it goes through several bench steps before you select the right reagent cartridge to load onto MERS-scope machine. And then the machine just takes about 30 minutes or so to set up, and then it's all fully automated downstream. And then it generates data, and then you can use the MERS-scope visualizer software to see the data. So the MERFish has three key steps. The first is to sort of hybridize with the set of probes, and then we do tissue clearing to remove the background, and then we do sequential image to sort of capture the barcode, and then do decoding. Then we understand which gene is which and at which location. So in the next few slides, I'm going to very briefly walk through some of the key advantages of MERS-scope and MERFish. And this is sort of a data really showing that MERFish is able to profile the gene expression in situ from whole tissue to subcellular resolution. And this is a human ovarian cancer example highlighted here with a few genes here with the animation on the right, highlighting all 500 genes at zoom in level and what the raw data looks like. So very briefly, some of the key advantages of the platform. First, it's a very quantitative and accurate measurement. You can benchmark with traditional technologies such as bulk RNA-seq or single molecule fish with highly expressed genes detected high by MERFish and not expressed genes detected low. And it spans many orders of magnitude here. And in high quality samples, there has been quite a lot of comparison papers of different spatial transcriptomic technologies. This is coming from Rahul Tikta's lab from New York Genome Center, published earlier this year in eLife, highlighting MERS-scope has the best sensitivity and most optimal trade-off between sensitivity and specificity across different platforms. This is also platform has very high throughput. Now it can scan up to 3 square centimeter versus the first generation of MERS-scope launched in 2021. Now it's able to have 2 times faster imaging. It enables true single-cell atlas, and this science paper is, I would call it one of the seminal papers describing you can use a targeted gene panel to be able to basically accomplish cell atlas in the Marcellus region from Xiaowei Zhuang's lab, demonstrated in 2018. And then, even though it's a targeted gene panel, you can integrate with single-cell RNA-seq or single-nucleus RNA-seq data and then get transcriptome-wide spatial data so that you can predict where the locations of those genes not measured from MERFish panel. And with that type of information, you can start to do ligand and receptor interaction analysis with special contexts. For example, highlighted here, a pair of ligand and receptor interactions in the mouse brain, highlighted D2-MSM and cholinergic neuron interactions here. Now you can really start to visualize this in situ. And then the platform is also compatible with protein co-staining. Highlighted here on the left is the strip protein stains and overlay of the protein, the transcriptome image of 347 genes on top. And as some of you may already know, recently Vizgen have already merged with Alti, which is a special multiomics company. We're quite excited about the next phase of special multiomics capabilities. And then this technology has been widely demonstrated in many different tissue and sample types with more than 270 peer-reviewed publications and preprints compatible with cells, frozen samples, and FFP tissue blocks. And then we're actually introducing MERFish 2.0 very soon, making the best even better. Basically, in some samples where RNA is degraded, MERFish 1.0 chemistry-wise, the sensitivity may be compromised. Now we're introducing MERFish 2.0 to increase the sensitivity, especially for degraded RNA samples. This will enable detection of RNA transcripts in more challenging samples, improving the differentiation gene expression. And with that, then you don't have to filter out so many cells doing single cell analysis and reducing cell dropout. And highlighted on the here, hopefully you can see adjacent FFP human colon samples, colorectal samples with Murphy's 2.0 at the bottom. You are able to capture way more cells, and then these are now more sort of accurate cell-cell interaction analysis. And these are just a couple of examples where our customers used Murphy's 2.0 and compared with Murphy's 1.0 in FFP samples. You can see in colorectal cancer sample with a few selected genes, you can see the improved sensitivity, quite many substantial improvement in terms of sensitivity here. And then this is in mouse skin, very similar story, almost 5 to 7-fold of improvement across quite a few samples. And then it just very last 10 seconds or so, just to highlight some of the key applications that we have seen using Murphy's technology. We have been seeing cell atlas in mouse, non-human primates, and the human brain using Merscope and Murphy's technology. And similarly, in oncology, we have seen a lot of studies in cell atlas in understanding the mechanism of immunotherapy and biomarker discovery and novel therapeutic targets for a lot of cool publications. If you're interested, highly recommend to read. Yep, and I will probably just stop here and just this is a quick slide summarizing all that I talked about. Hopefully gives a sort of sense of what Merscope capabilities.
Jiang, thank you so very much. And I'm really glad to see more teams embracing this technology that gets us better data for the multiscale atlas. So thank you for developing it, making it ever more sensitive and robust. Let's go over to the next presenter here in the interest of time. So next up is Colles from Takeda. Are you okay to run your own slides?
Yeah. I will share my
Dr.
screen.
Price.
Let's see. Hopefully everyone can see.
I can see it, not yet in presenter mode.
Is it presenter mode?
Not yet on my end.
Do you see the slides though? Anything?
Yeah, I do.
Okay, perfect. We'll just go. Sake of time, we'll just start. So thanks. I wanted to thank everyone for the opportunity here. My name is Dr. Colles Price. I run spatial omics and spatial genomics over at Takeda. I really was going to just for a few minutes just introduce some of this idea of spatial mapping, ways to analyze, ways to improve stuff, as well as new machine learning and building capabilities. So really this early idea of systems biology came from this paper from the early 2000s, Can a Biologist Fix a Radio? So it's a great paper. I very much recommend everyone to check it out. But ultimately the idea is that a biologist, we take an almost systematic, almost reductionist approach to approaching problems, where we would kind of just knock things out or disrupt things one thing at a time and build a signaling pathway that way. This should look pretty similar to a lot of other pathways you might see out there, and it leads to some sort of phenotype. However, biology is more complicated, right? And it's like really, if we think about circuitry of biological cells, this is true for whether you were looking at things like radio or things that are like in the biological context of tissues and homeostasis. And really, this is important because, you know, as the technologies are evolving, as we just heard from the last two speakers, as well as other speakers, we're really shifting from bulk to the single cell approach, but the context in spatial matters. And really, we've been trying to move from the single cell approach to spatial approach and understanding how things interact with each other on this individual cell basis. You could use this for things like tissue atlasing. You could look at cell-cell interactions, build neighborhoods. You could look at cell signaling, ligand receptor mapping, and molecular interactions.
Would you like us to see other slides? We still see the cover slide.
Oh, no.
All right.
Let's try this again.
And we will make all these slides available. I could
Yeah.
also run them if you prefer. I think I can do this relatively quickly. Thanks for sharing all these slides in that giant deck.
Yeah,
I can share my screen if you like. Okay. Maybe you just have to hit the presenter mode. Yes, now you're in presenter mode. Perfect.
Okay.
Thank you.
And so we could talk about this with sequencing-based approach, imaging-based approach. There's pros and cons to multiple ways of doing spatial transcriptomics, but for the sake of time, I'm just going to introduce a little bit of data from both. And so this is generally what you'll see from a lot of spatial omics people. You do single cell imaging-based approach, you generate your UMAP, and now you can project your UMAP onto your tissue of interest. You can look at cells within the tissue, individual transcripts or genes or cells within and across the tissue. As you can see, different expression of these genes in the tissue here. You can also see where it is in the UMAP to probably have indication of how specific that gene expression signaling is. We can build it to stratify patients. This is a story that we published last year, where we saw that a lot of patients treated with immune checkpoint blockade hepatocellular carcinomas, despite having rich T cell environments, did not really respond to therapy. Ultimately, using spatial transcriptomics, we saw that it wasn't just the presence of immune cells in these hubs, but really it was the composition of the hubs. And we found essentially it was this MREG-DC CXCL13 TH-like cluster that was best predictive of therapy. And so much so that there's follow-up studies looking at new clinical trials using this information to stratify patients. You can also, as I mentioned before, build this into different types of neighborhoods. And there's a few ways to do this. For the sake of time, I'm just going to introduce a couple quick ideas about neighborhood analysis. You can build neighborhoods based on defining regions of your tissue. You can define small regions, you can draw large regions. In circle here, each one is a cell type. As you can imagine, the bigger region you define, the more cells you're going to capture within that neighborhood. I compare this to thinking about New York City, right? We know we're looking at Upper Manhattan, Bronx, Brooklyn, and Harlem, and can see different types of populations and different composers of different areas in New York City. So here, this is looking at MERFISH data. We took the similar approach by applying tiles to the MERFISH data, and then we could use those tiles. We can UMAP on the tiles itself to see regions and neighborhoods that are very similar to each other. We also published this paper earlier this year, focused on that same idea, where we were able to build up these spatial neighborhoods as a function of tiles. And ultimately, we were able to find tiles that were better predictive of clinical response, that we didn't see the same thing with single cell sequencing data. So it was very specific to the tiles. Another way, instead of defining the neighborhoods by Brooklyn, Manhattan, Bronx, you could look at demographics of New York City, right? Like, where are the Latino populations, the Black populations, the white populations, the Asian populations? And so from there, it builds a very different type of neighborhood and a very different type of map. We used to do this, we worked with Nick Fernandez to build something called alpha shape approach. And so the idea is that you set a distance threshold for your cell types. And if you hear in this example here, you can see there's some cells in the same cell type that are too far away and so aren't built into this alpha shape. However, these other cells within a certain distance are built into an alpha shape. And this is quantifiable. You can quantify how big the shape is, how many shapes it formed, and how shapes overlap with each other. Again, choose these type of approaches require choosing the right size. Here we see that our size is really, really small, so there's actually not many shapes being formed. Here we see our size is too big, and so everything looks like a big jumbled mess. But when you find that right size, you can even start to see true pathology. So here we can see endothelial cells of vessels very clearly here in yellow throughout the tissue. We start to see other fibroblasts and areas of interest, and it's very easy to detect. So for the last minute, I'm going to talk a little bit about the sequencing-based approaches, because they have made a lot of improvement over the years. We've also used Visium HD, which you can see Visium compared to Visium HD is dramatically better with the increased tile number, with reduction in tile size, to make much higher resolution data compared to previous sequencing studies. We've also used LightSeq, which broke out another lab at Harvard, And here it uses optical barcoding where you select your cells of interest on your tissue, you flash light to tag the cells, and you could do this multiple times with different types of markers or cells. But once all your cells of interest are tagged, you collect them, sequence them, and now you can get really, really high resolution. They demonstrated this in this paper that came out a couple of years ago, where they were able to tag and isolate 6 dopamine anchored cells, which there's only a few per section, and get new tissue biology. Ultimately, biology is complex. It's beautiful. It's awesome. But really building in these models, I think, is really important. And really, with the new technologies, I encourage everyone to open your mind and really drive new questions. And I'll end on one of my favorite quotes. You want your technology limited by the question you ask, not the question you ask limited by your technology. And this is my quote I said a couple of years ago. And with that, I'd like to thank everybody. I think I'm right at 8 minutes, so I'll stop sharing now.
Thank you very much. Yeah, whenever you hear the loud noise, that's the ending of it. Thank you for presenting, Dr. Colles. Nice to have you here. Next in line is Daniel Moline from 10x Genomics, and I'm so glad you could join us all here today. Many of us are using your technologies. And if you want to share, then we can see more of your work. And Dan, thanks for setting the timer.
All right, so everyone can see my slides, right?
Yeah.
Awesome, great. Okay, so I'll just dive into things quickly to try to keep on time. So just as a brief introduction, my name is Dan Moline. I'm a science and technology advisor with 10x Genomics. And what I'm going to talk about today is kind of the entire portfolio that we offer, and kind of just to describe through a preprint that we have available, kind of how integrating these technologies together can actually yield some really powerful insights, especially when it comes to profiling deeply specific cell populations. So just to briefly describe the portfolio, we have 3 kind of main platforms that we offer. And so Chromium single cell is probably the one everyone's most familiar with. That's single cell RNA sequencing with a very varied group of modalities you could add on top of that single cell gene expression information. And we also offer Visium Spatial that Dr. Price was just describing in a few slides ago, allowing for whole transcriptome profiling while maintaining that spatial context. And then last, we offer Xenium In Situ, which is a multiplexed in situ platform with an imaging-based readout, allowing for really fast, really sensitive in-depth profiling, while also allowing for a subcellular resolution in those resulting data. And so the publication that I wanted to describe here was one where our internal scientists kind of wanted to put these platforms through their paces and use them on some patient samples isolated from colorectal cancer patients. And so this is kind of a disease state focused paper, but what it allowed for was kind of the integration of these 3 different platforms together. And so sections were taken from FFPE blocks and run on the Visium CytAssist platform, allowing for Visium HD profiling, as well as single cells were isolated from these FFPE sections, allowing for single cell RNA sequencing of cells from the FFPE block. And then last sections were also put onto the Xenium analyzer as well. And so first among these kind of profiling approaches was the Visium HD platform. And what that allowed for was a whole transcriptome analysis of the tissue with spatial context maintained. And so the feature size on the capture areas of the Visium slide is 2 micron by 2 micron, allowing for a single cell resolution of those transcriptomic outputs. And the CytAssist platform is what's used for transferring analytes from tissue onto the Visium slide. So there's no need for sectioning tissue onto the Visium slide. And when you put tissue onto the CytAssist, you can use things like archival samples that have been hardcover slipped even for running on the CytAssist. So you can kind of dig into those biobanks that you have available of tissue to then profile it downstream. And so the output much like what Dr. Price already showed, when compared to the original Vizgen platform with its 55 micron features, is much, much finer, where these really dense and complex mucosal tissues could be resolved, showing not only the morphological information contained, but also heterogeneity within those regions. You can see that there's a massive increase in the number of clusters and the diversity of cell types present and viewable in these tissues in the HD platform when compared to the SD platform. But kind of a natural obstacle that can happen with these spatial platforms is that because they're so new, identification of cell types can kind of be something that needs extra work. And so what can be nice is actually leaning on single cell data that's already been generated with this platform that's been around for almost a decade now, to use it as kind of like this ground truth that you can use to help impose those identifications you've already worked so hard to make in single cell onto your spatial data. And that's actually what they were able to do because they were able to get the fixed RNA information from the exact same tissue block. And what that allowed for was the identification of a really, really wide and diverse number of cell types, where up here is just the spatial information, and here is the information that's used integrating single cell and spatial. And not only did that allow for the identification of a wide variety of cells, it also allowed for the removal of some of the features that sat under multiple cells on the slide. Since the features are kind of immovable, they're in that capture area. When you had that fixed cell information, it allowed for a differentiation of high quality features from low quality ones, having a really fine, well QC'd data set for identification of cells. And those cell identities were then used to analyze the periphery of the tumor, kind of this region with really dense and diverse cell types, this microenvironment that's kind of at the center of a lot of the in-depth cell profiling that's in the cancer field. And what they wanted to do was look at these regions that are roughly 50 microns away from the tumor edge and analyze the cell types within it. And their identification of cells is something that's not really shocking. It was mostly macrophages and cancer-associated fibroblasts. But what they wanted to do was profile the macrophages and see if there were some interesting cell populations within. And what they found was that there were two separate macrophage populations, one high expressing in SBP1, another high expressing in selinop. And what they found was that these populations not only localized distinctly from one another, shown here in panel C, but also that the adjacent tumor cells were expressing differing oncogenic programs depending on which macrophage population they were next to. So we didn't just find diversity here, we were also able to imply a sort of interaction that wouldn't be possible with other platforms. And lastly, one of the things that's really frustrating about these really dense and highly diverse areas is that you can kind of reach the limitations of resolution that are possible on whole transcriptome platforms. So we also employed Xenium in this preprint to kind of understand more deeply these regions at the periphery. And this platform is made to be kind of this very easy to use, highly customizable platform that allows for high resolution profiling and kind of a quick turnaround. And so what was done was not only did we analyze the T cells in these regions, which are highly rare cells, very difficult to find in these regions. We can see that they're analyzable and detectable with Visium HD, but in-depth profiling could be somewhat difficult. Not only do we find these cells and find the neighboring cells to kind of understand the cell-cell interactions in this region, we were also able to profile specific known T cell receptor sequences that are known to prime those cells to attack tumor cells. So we were able to really in-depth profile the possible anti-tumor response of the immune system in the microenvironment in a way that's not really possible on other platforms. And just to gently push back on my colleague Jiang's assertions about MERSCOPE, there are other side-by-side studies, including this one, which compared over 100 different samples that varied greatly in tissue source and disease state, found that Xenium was actually at least 2 times more sensitive than MERSCOPE. And specifically, this image I think is quite telling. Xenium is much more robust to variances in sample quality, which is the natural state that almost everyone deals with every day in science. And ultimately, Xenium was able to provide higher quality data from these sets of samples than MERSCOPE was. You can just see the comparison here where Xenium has much more high quality information. And so I just wanted to kind of finish up by saying thanks everybody for inviting me and to kind of encourage folks to reach out to reps, not just from 10x, but everybody to find what technology is the best fit for you. Because ultimately it's a pretty complex state of play here in biotech. And we're more than happy to kind of make sure that everybody is pursuing platforms that fit best with their biological questions. So thanks, everybody.
Thank you, Daniel. Great timing also. It's wonderful to see these algorithm comparisons. I think we absolutely want comparisons of technologies, of algorithms, of methods, of data visualizations, of different layout embeddings, etc. So this is right in line of what science is all about, being reproducible, transparent, comparable. And so thank you for bringing this up. And I hope to see some of the human cell atlas data and also the human reference atlas data used in these comparisons. Next up is Sarah Teichmann. She is at the Cambridge Stem Cell Institute in the UK, joining us, I think, not from there, but from some other place on this planet, not on Mars yet. And she is also a CIFAR co-director, and it's a real pleasure to have her here. Thank you for presenting, Sarah.
Hi, everybody. Can you hear me?
Yeah, we can hear you, and we see your non-presenter mode. Now the presenter mode, wonderful, thank you.
Okay. So I'm going to take a step back from the technologies and kind of give an overview of where the whole community is at right now in terms of mapping the human body at a very coarse-grained level through multiple different technologies. And so we've just heard kind of the in-depth deep dives into some of the single-cell and spatial technologies that are being used in the community, both for studying human, but also other samples from mouse and zebrafish and other model organisms and so on. And, you know, what those presentations really highlight is what the potential of these technologies for a deeper understanding, in particular, what I'm interested in is the human body, in other words, ourselves. So imagine what we could do if we had these high-resolution, detailed cellular maps with full molecular breadth for all 37 trillion cells in the human body. That's obviously a huge task, and it's not something that a single lab or even institute can tackle. So in order to achieve that map of the human body, we need to work together. And what can such a map of our cells at the molecular level unleash? Well, it could allow us to, for instance, engineer cells in the dish that are faithful to our cells in vivo at a level of molecular detail that's unprecedented, and sort of benchmark and develop human cells and tissues in vitro. It would also allow us to map individual genetic variants, individual drug targets, individual pathogen entry factors.
We only see the blue screen. We don't see your slides.
Yeah, no, it's just introduction.
Oh, my bad, sorry.
Yeah. The technologies that enable us to do this are single-cell genomics, which you've just heard about, which was first sort of in my mind kind of first published in 2009 by Azim Sorani and colleagues in a manual approach mapping 5 individual cells in the developing mouse embryo. Now, of course, there are many high-throughput commercial technologies, and you've heard about some of them. The challenges are handling the human samples, the genomics of sequencing the individual cells or nuclei, and then the data analysis for identifying the different clusters, which some of you have alluded to briefly. And obviously this is kind of enabled through the power of AI/ML, statistics and computational methods, which are kind of at the heart of this endeavor. The spatial technologies that you've heard about are kind of tightly coupled to the single cell. I'll just activate my laser pointer, and of course the sort of imputation and deconvolution of the geography of gene expression and molecular landscapes of tissue samples basically then tells us about those niches that Dr. Price referred to, and that help us understand our tissue architecture both in health and in disease. And the project basically to create an assembly of cell atlases for the different tissues in our body is the Human Cell Atlas Project. And we, with Aviv Regev and many others, we sort of assembled the community back in 2016 towards the endeavor of mapping the cells in our body. And there are now over 100 institutes and over 3,000 members that contribute to this project and that are registered towards this endeavor in over 100 countries around the world. And the cell atlas is sort of the level of cellular resolution that we've been discussing in this session. Now, of course, in future, this layer will sort of fit into larger volumetric kind of technologies all the way up to imaging and technologies like the X-ray scattering that you see here, medical imaging and so on, and down into subcellular resolution, like, for instance, electron tomographic reconstructions of individual cells and so on. One example of the types of information that that's been incredibly inspiring to me is resolution of rare cell types like the pacemaker cells in our heart that are in this animation. And it's really the molecular data that's allowed us to understand those cells that set the pace of the beating of our heart at a level of detail that wasn't possible previously. Another example is mapping the viral entry factors across the different tissues, which was a sort of emergency project that came together in the pandemic where we were all under pressure to understand how the virus enters and how it's transmitted between individuals. So in early 2020, we sort of put together all the draft data in the Human Cell Atlas community in a kind of exemplary, massive, speedy collaboration. And what this revealed was cells particularly in the eyes, cornea and conjunctiva, in the nasal epithelium, in the oral cavity, in the ductal epithelial cells in the salivary glands that harbor the viral entry factors ACE2 and TMPRSS2. And this detailed cell and molecular information helped us understand where the virus is entering and, of course, the dynamics of transmission, and informed public health policies around mask wearing and protective equipment and so on. So you can see the power of this very detailed sort of Google Maps of the human body. And if we take this into the dish, into engineering of cells, then it's obvious that the Human Cell Atlas is giving us the recipe for the factors that are needed to engineer cells, both cell intrinsically, but also in the tissue niche, in terms of extrinsic factors that are signaling to cells. And we've heard from cell signaling technologies, this intraseq technology that tells us about the protein signaling pathways. And if you take we stare into a crystal ball and kind of unleash our imagination a bit further, what this would allow us to do is to also, once we can synthesize DNA synthetically, the way it's been done for yeast genomes, for mammalian genomes, we could then dial a chromosome, for instance, or a set of human chromosomes that have the genes that are needed for a particular cell state that has functions that we wish, that has particularly regulatory elements in non-coding regions and excludes all other cell functions. And so we could sort of, using the data about genes, their regulatory elements and gene modules, we can imagine the power of the Human Cell Atlas for informing synthetic genomics. So the future is incredibly exciting. I just want to finish by saying thank you to the Canadian Institute for Advanced Research Multiscale Human Program, that Katy and I and Gary Bader are co-directors, and of course handing over to Tobias, who's an Israeli scholar of that program. And thanks Katy for the opportunity to speak in this session.
Thank you for the exciting vision. It's a little scary also, but it's also mostly exciting. I think that's really doing that all can go wrong also. All right, Tobias, please. It's good to see you here. And I know you're joining us from Chile, right? Mm-hmm.
Yes, exactly. So far away from some of you, it's quite an international and time zone spanning event. So thanks again for the invitation, especially because in contrast to many of you, I don't have a lot of exciting big data sets to share yet, but mainly methodological approaches that are new. And it fits really well because we've already heard about a lot of microfluidics in the previous presentations, even if not talking step by step about the microfluidics methodology. Still, most of these equipments are based on microfluidics. Even sequencers are essentially microfluidics and flow cytometers. And so when implementing new methods, researchers usually have a choice if either they can find a commercial instrument and maybe reagent kits to implement the methods quickly, or to build the whole thing themselves, which gives you new opportunities to combine things in a different way and do new assays. And of course, it's also cheaper, but more difficult. So a lot of the things we've seen here earlier today would not be any option for my research group in Chile because they're just too expensive and it's too difficult to get all the kits here. So I will talk about a new kind of assays or approaches, talking about the interactions of cells. So we've already heard how spatial omics can help to see where cells are co-located, and then you can derive hypotheses. But in the end, you still need to perform in vitro experiments to really see how things interact. And sometimes you want to see other scales. So you might want to see how different organs interact, for example, through, for example, organoid models. And you don't get that by seeing what's next to each other on a microscale. So I'll talk about that. And how also, I already mentioned organoids, so having little model organs and see how they behave in vitro. These can also be improved with microfluidics, so they can be made more reproducible and having more experiments in parallel with microfluidics, and how we aim to make all of this methodology more accessible. So, as I said, we've already heard a lot about microfluidics. It's a very versatile method, in particular droplet microfluidics, which has really been helping with single-cell genomics also to create this environment where different reagents or DNA molecules are sort of trapped and can be manipulated and barcoded. So I'm just jumping over this quickly. The slides will be shared. This is also not our publication, but a reference. And droplets are really great also to look at interactions. So for single-cell genomics, for example, transcriptomics, we use molecular interactions in these droplets to barcode, for example, individual transcripts. But you can also use this for metabolic interactions between cells or, for example, tissues and bacteria. So I also have some background in microbiome studies, and this is an interesting layer of complexity that I'm also interested in. Because the advantage is that you have the separate environment, so different parallel experiments don't interfere with each other. They're all divided by oil, but inside the droplet, there's quite a low volume. So if you use patient samples, for example, they are diverse in terms of cell types and expression profiles, then if you want to study them in their interactions, you basically need to dilute them. So you often start out with a single cell or very few cells. And so when you put them into a normal well plate, let's say, even if you try to use small volumes, these volumes are gigantic for the few molecules a single cell might produce, and they get lost in space, and you have a high risk of contamination. But in a droplet, these interactions happen really fast and they're contained. So droplets are really great for studying interactions. And in fact, we published this review last week with some really interesting methods across different types of interactions of proteins, molecules, and cell types that are mainly from the last 2 years of publication. So it's a really interesting field. And one of the things that we are interested in and starting to do in my lab, so this is not yet data from our lab, but another reference, you can sort of merge different droplets that have different types of molecules and cells, for example, in this case, bacteria and spheroids, but we work with more differentiated organoids. And you can then study the interactions between these two partners, for example, and you can image them over time to understand how this interaction evolves. And so these are some of the first organoids that we're working with. Here's some light sheet 3D data on the organoid. So it would be nice to integrate some light sheet also into these assays. So we're building all this equipment ourselves in this case. So organoids are these little tissue balls, essentially. And the difference between spheroids, I mentioned them already, and organoids, I don't know who else is listening, so I'm just going to try to explain this in a basic way, is that organoids are differentiated. So usually start with stem cells that in our case, we derive from patient samples. So you start with a patient biopsy to have the genomic individual characteristic represented. But then from these cells, you derive a stem cell line that might take a week or so at least. So it is a bit of a lengthy protocol. You need to sort of grow some of these cells first until you have enough. And then from these stem cells, you grow these little sort of tissue balls, and they differentiate with different cell types that are typical for this tissue, and you guide this with sort of different chemical markers to achieve this differentiation. And then you can use these little balls again in microfluidic droplets to represent the tissue and see how that tissue interacts to certain stimuli or interactions with other cells or even other organoid types, other tissue types. And one interesting technology that has really enabled single-cell transcriptomics, but also in this case organoid studies, is actually making little microgels, which is a little bit more challenging than just making normal liquid droplets in oil. And we made this open source workstation with which you can make these, for example, agarose-based oil droplets in oil, microgels in oil, by introducing sort of different temperature control. And in order to make this equipment more accessible and so that more labs around the world can perform these experiments, we made this open source, the design, but it's also really low cost. So it's based on a Raspberry Pi, and it's all based on digital fabrication. So you can 3D print most of the parts, laser cut some of them, but you can also 3D print those parts that we usually laser cut for convenience. But it's all based on digital files. So we share the files, and it's really easy to change how this looks, or if you need to incorporate something else, because you have these files available and you can just print the variation. And there's a step-by-step IKEA style, let's say, documentation available. So really anyone without hardware knowledge can replicate this at home or in a lab. We run different workshops also, including an EMBO advanced training course of that's related to metagenomics, and different workshops, especially across Latin America, to give different research groups access to this kind of equipment and to start using these workflows. Like we discussed in this meeting, most of these workflows are not done by many groups across Latin America, and therefore the biological diversity is also not well represented in these atlases. If you're interested in more approaches of how to make equipment more accessible, open source equipment, there's an article from me also that you can read upon, because here in the interest of time, it's just a very brief summary, and we have a lot of training videos and 3D printed files that we share also available online if you want to check it out more. So thank you very much for having me with this different perspective. And yeah, with that, I hand back over to Katy.
Thank you so very much. Perfect timing, Tobias. And I did pose a question to everybody, especially those which produce technologies. Are there special programs that people from Latin America or Africa could benefit from to try out the new technology and make sure that it's really an equitable atlas in terms of data used, but also in terms of people contributing to it?
So just to describe from 10x's perspective, typically when people want to contribute to the atlases from countries like Africa or Southeast Asian countries, we leverage the Flex platform. So we use their collaboration with people in countries that have easier access to the instruments to then send fixed samples intercontinentally. So that way, then we can make sure that the data is still high quality, that everyone gets what they need, but they don't have to try to acquire and create lab space for an instrument if they don't want to.
Yeah, I think this, especially the sort of workhorse 10x instruments, have had an important role to play in this methodology space, I think, because they really provide very replicable, sort of high quality data. So I think with these custom methods that you can build maybe it's more difficult to maintain very comparable data between different labs, or at least that's something where an approach needs to be developed to manage that. So that's why there are also some of these instruments used in Latin America. But as far as I know, the groups that use them mainly survive on international funding. So especially CZI has been really helpful in pushing this and providing, and exactly according to this question. So having in mind that we need representatives across the world, they financed some groups to do this. But I have my doubts how well they will be able to continue this work if the CZI funding ends. So it would be great if other funders would take that in mind. Here, there's not so much collaboration between Latin American countries to share funding. So there's not European funding or something like this available. So there are just very few foundations and funding sources that can be used.
Well, one thing that I've been pushing for the last year or so is not necessarily instruments and access to instruments, because that requires like a long infrastructure investment funding wise, but with training, upkeep. But I've been really pushing very strongly for making kind of like the Human Cell Atlas and HubMap, making kind of an open source almost data posting and sharing. Like all the companies, we have so much data sometimes that we just don't have the resource, the personnel to answer all the questions we're interested in. So if NIH had funded like a more different types of spatial atlases where we could share that data, that makes it a little bit more accessible in the sense that ideally anyone in the world could use that data and analyze it. And so that's slowly, I think, making a little bit more leeway, at least now than close to a year ago. But I think that's something that would make things a little bit more globally accessible off the bat.
Yeah, in the funders panel yesterday, we also had quite a few panelists which have now joined programs around the globe. So maybe check that out as well, Tobias. Everybody, thank you so very, very much for being on this panel. It's a true pleasure. I hope to collaborate more over time. Please also talk to each other. I think it's always good to compare notes. And of course, there are many events that bring us together, so maybe we can see each other in person also. Very good. Thank you so much. Dan, you're going to bring in everybody else for the 11 o'clock panel, and then everybody else, feel free to stay on as attendees. We love to have you. Thank you.